Hey everybody, welcome to chapter 5, lecture 6. In this lecture, we're going to be talking all about cell membranes, their structure and their functions. They're going to be divided into three specific videos. The first one is going to talk about the structure and the components of the cell membrane. The second one is going to talk about passive transport or diffusion. And the third one is going to talk about active transport. Enjoy. The plasma membrane or cell membrane serves several functions. First, it acts as the outer membrane, outer border of both cells and their organelles. As a border, it regulates what enters and exits, ensuring only necessary molecules get in or out. Secondly, it plays a role in receiving signals, from external environment. These signals can trigger responses inside the cell. Think of it like a cellular security guard. It monitors incoming signals and initiates actions when needed. Lastly, the plasma membrane helps with adhesion. The plasma membrane helps adhere to their neighboring cells, allowing them to stick together. We discussed this in previous lectures when we talked about tight junctions, desmosomes, and gap junctions, which enable cell-to-cell interaction and communication. While we previously described the cellular membrane as primarily composed of phospholipids arranged in a bilayer, it's essential to recognize that the membrane is more complex than that. We refer to it as a fluid mosaic model because it is not just made of phospholipids. It's also composed of cholesterol, proteins, and carbohydrates. The term fluid indicates that these components aren't fixed in one place. They can move laterally, like people in a busy crowd. This fluidity allows the membrane to remain flexible and functional. Researchers discovered the membrane's bilayer structure in the 1950s, thanks to the advancement of transmission electron microscope. This tool enabled scientists to visualize that the membrane was composed of two layers of phospholipids, not just one as previously thought. In general, the plasma membrane is about 5 to 10 nanometers thick. For a typical cell, 50% of the membrane is made of proteins, 40% is composed of lipids, and the remaining 10% consists on carbohydrates. Although these percentages can vary depending on the cell type. The carbohydrates on the surface of the cell membrane can attach to proteins, forming glycoproteins, or to lipids, forming glycolipids. Phospholipids are the primary component of the cell membrane. They are amphipathic or amphiphilic, meaning that they have both water-loving hydrophilic and water-fearing hydrophobic parts. Let's break them down. The hydrophobic tail are two fatty acid chains that are non-polar and do not interact well with water. The hydrophilic head is made of glycerol attached to a phosphate group, and sometimes other attachments like carbohydrates. The fatty acid tails can be either saturated or unsaturated. Saturated, of course, meaning that they only have single bonds between their carbons, and unsaturated, meaning that they have at least one double bond, which gives the tail a kink. These kinks make the membrane more fluid in regions with unsaturated fatty acid tails, while the saturated tails pack more tightly, making those regions more solid. When many phospholipids gather, they naturally arrange themselves into structures based on their amphipathic properties. Bilayer sheets, bilayer sheets. This is a structure we typically see in membranes, where the hydrophilic heads, these white heads, face the outside of the cell and the inside of the cell, while the hydrophobic tails face inwards. Liposomes, shown here, are smaller spherical structures formed by a bilayer. Both the inner and the outer surfaces of the liposomes are lined with polar heads or hydrophilic heads, while the tails remain in the middle. Lastly, we have micelles. These are also spherical structures, but consist of a single layer of phospholipids, where the hydrophobic tail points inwards, usually trapping something in the inside, something hydrophobic, and the hydrophilic heads face the outward. Proteins are vital to membrane function. They serve several roles. They can be transporters, which move substances across the membrane. They can be receptors. They bind to signals like hormones and initiate cell response in the inside of the cell. They can be enzymes that they speed up reactions on or near the membrane. They can be adhesion proteins. They help stick to surfaces or other cells. Membrane proteins come in two types: peripheral or integral. Peripheral proteins are located on either inner or outer surface of the membrane. Integral proteins are embedded within the membrane. They're embedded within the membrane, often spanning both sides of the phospholipid bilayer. Again, the peripheral proteins are attached to the inside or to the outside of the membrane, but they do not cover the whole membrane. While the integral proteins can go through one side to the other side of the protein. I mean, of the cell membrane. Integral membrane proteins need to interact with both the polar region and the non-polar region of the phospholipid bilayer. In this picture, let's assume that these are our integral proteins. Part of the integral proteins will be dealing with the hydrophilic side of the bilayer, while the another side will be dealing with the hydrophobic side of the lipid bilayer. Therefore, they have a hydrophilic region of their own and a hydrophobic region of their own. The hydrophilic region will interact with the outside, while the hydrophobic region will be interacting with the inside. So let's consider a structure of the structure of an integral membrane protein. So for that, let's start doing some drawings. Personally, drawings help me a lot to understand these concepts. So these are the phospho heads, and now I'm drawing the hydrophobic regions. And here comes our membrane protein. Let's say, yes, like that. And here comes the rest of the phospholipid bilayer. Okay, now, if I were to guess which amino acids are found in the inside part of the membrane protein and the outside of the protein, remember, we're made of 20 amino acids. Some of them are hydrophobic, like these ones over here, and some of them are hydrophilic, like these ones over here. So, if I were to guess which proteins go where, which of these proteins, where do you think the lysine will go? So first of all, what is lysine? Lysine is positively charged, so most likely can interact with H2O, right? So lysine can go on the outside. But then, what if I give you leucine? Oops, that's lysine as well. What if I give you leucine? Leucine is a non-polar. It is a hydrophobic amino acid, so it most likely will go on the inside of the protein, away from the water. So for the examinations that you want to have for the test, I will suggest you to remember that I will suggest you to remember how does the structure of the amino acid gives it its the R group, gives them the chemical properties of hydrophobic and hydrophilic, and where they should go in a integral membrane protein. The third major component of the plasma membrane are carbohydrates, especially oligosaccharides. Remember that oligo means few. So these are short chains of sugar or short chains of monosaccharides. You'll always find these carbohydrates on the exterior surface of the cell membrane, where they are bound to proteins to form glycoproteins, or to lipids to form glycolipids. For example, you can see in this diagram a glycoprotein. Let me find the glycoprotein right here, and a glycolipid right here. These molecules play a crucial role in cell-to-cell cognition and attachment. A great real-world example is our blood type. I'm sure that you all have heard of blood types A, B, AB, or O. It's actually the type of glycoprotein found on the surface of the red blood cells that determines what blood type you are. For example, blood type individuals has A glycoprotein, while type B has B glycoproteins. Type AB have both A and B glycoproteins, while O does not have any of these A or B glycoproteins. While glycoproteins usually help the cell function, there are some exemptions. For example, in the immune system, we have T cells. Here are T cells over here. This is a cell membrane of a T cell. And B cells. T cells have a glycoprotein called CD4. This CD4 is a receptor on the cell surface. But unfortunately, it is recognized by the HIV virus. Instead of recognizing HIV as a harmful agent, the CD4 receptor mistakenly begins to interact with the HIV virus, allowing the HIV virus to enter the cell. We will go deeper into this when we cover the chapter on viruses towards the end of the semester. Membrane fluidity is critical because the membrane needs to be flexible. The membrane needs to be flexible to prevent tearing, but also rigid enough to maintain its structure. Imagine the membrane as a sort of cellular Goldilocks zone, not too solid, not too fluid. There are three main factors that affect membrane fluidity: phospholipid type, where saturated fatty acids, or SFA, make the membrane more rigid, and the unsaturated fatty acids create more fluidity because they do not get packed as tightly as the saturated fatty acids. Temperature also plays a role. Warmer temperatures increase membrane fluidity, while colder temperatures make it more rigid as the molecules compress. Cholesterol presence in the membrane also have a role. Cholesterol is like a buffer for the membrane. It helps maintain fluidity in cold conditions, like during winter months, keeping the membrane from becoming too rigid, but also increases rigidity during warmer conditions to prevent the cell membrane to become too fluid in the warmer months. The plasma membrane is also asymmetric, meaning that the interior, the cytoplasmic inside, and the exterior, the, the area that is faces the extracellular environment, are not the same. The interior proteins are often anchored by cytoskeleton, which includes microtubules, microfilaments, and intermediate filaments. The exterior proteins interact with the extracellular matrix, which helps connect the cell to its environment. Glycoproteins bind Glycoproteins help the surface to bind to hormones and other signaling molecules, allowing the cell to respond to its external environment. Because of the composition of the membrane, primarily phospholipids, the plasma membrane is selectively permeable. This means that most substances cannot pass through, but some molecules can. This selectivity allows the cytosol, the fluid inside the cell, to differ in composition from the extracellular fluid. For example, the concentration of sodium and potassium are quite different inside versus the outside of the cell. The difference in concentration of sodium and potassium from the inside of the cell to the outside of the cell is going to be very important for many cellular processes. Now, in the next in the next video, we will talk about different types of transport across the cell membrane. One of them is the passive transport, which does not require energy for it to happen. And the other one is the active transport, which require energy like ATP for it to happen. All right, so this is the end of the first video. The next video will talk about passive transport or diffusion.